Photosynthesis is the process that plants use to turn carbon dioxide, water, and sunlight into their own food, while also producing oxygen as a waste product. This will make more sense after we go over the two main stages of photosynthesis: the light-dependent reactions and the Calvin cycle. Both of these take place in the chloroplast, the organelle in plant cells that performs photosynthesis. It has a double membrane, hollow-shaped donuts called thylakoids, which are organized into grana. The fluid-filled space surrounding thylakoids is called the stroma, and the fluid space inside the thylakoids is called the lumen. The light-dependent reactions, or also known as the light reactions, take place in the membrane of the thylakoid. Before I explain the process, let's understand the structure. This is the thylakoid membrane I mentioned, with the lumen inside and the stroma outside. Here is photosystem 1 and 2. They have pigments, called chlorophyll, which capture light energy. Right next to them is the electron transport chain, a group of proteins in the membrane that passes down electrons. At the bottom is ATP synthase, an enzyme that synthesizes or makes ATP from inorganic phosphate and ADP. Last is NADP+, an electron carrier. It carries and transfers electrons like a shipping truck. To understand NADP+, you need to understand redox reactions. Redox reactions is short for reduction-oxidation reactions. In reduction reactions, a substance gains electrons. NADP+ is reduced to NADPH when it gains electrons, while in oxidation, a substance loses electrons. NADPH can get oxidized back to NADP+ when it loses its electron. To help you remember, use the mnemonic Leo-Gair: loss electrons equals oxidation. Gain electrons equals reduction. Now the process starts when the chlorophyll in photosystem 2 absorbs sunlight, energizing or exciting to electrons. These electrons are then transferred to the ETC. To replace the 2 electrons in photosystem 2, photolysis occurs. In photolysis, an enzyme in photosystem 2 called the oxygen evolving complex splits water into protons, electrons, and oxygen. Electrons are donated to photosystem 2. Protons stay within the lumen, and oxygen is released as a byproduct or waste product because it doesn't serve any functional purpose for the plant. OEC actually splits 2 water molecules at a time, so at the end there will be 4 protons, 4 electrons, and 2 oxygen atoms. If you could recognize, this is an oxidation reaction because water is losing electrons. Now the electrons transferred through the ETC lose or release energy on the way. This energy is used to pump protons into the lumen from the stroma, creating a proton gradient, or a concentration gradient with protons. A concentration gradient is the difference in concentrations between 2 spaces. This area has a higher concentration than this area. Naturally, molecules go along the concentration gradient from high to low to create balance. This is what the protons will do. The protons will move from high to low through ATP synthase to power the making of ATP. This movement of protons down the membrane to create energy is called chemiosmosis. This ATP will be used in the next cycle. Now, after the ETC, the low-energy electrons will arrive at photosystem 1. The chlorophyll in photosystem 1 will absorb light, re-exciting the electrons. These electrons are then transferred to NADP+, creating NADPH. NADPH will also be used in the next cycle. Okay, so let's review. Photosystem 2 absorbs light, exciting electrons. Electrons are transferred to the ETC. To replace electrons, photolysis occurs, resulting in electrons that are donated to photosystem 2, protons that contribute to the proton gradient, and a byproduct of oxygen. Excited electrons go through the ETC, creating a proton gradient. Chemiosmosis occurs, and ATP is made. The electrons from ETC go to photosystem 1 to get excited again. These electrons are then picked up by NADP+ and become NADPH. In the end, you will have a product of oxygen, NADPH, and ATP. Two of them will go and will be used in the Calvin cycle. The Calvin cycle, or also known as the dark reactions, take place in the stroma. The cycle converts carbon dioxide into sugar in three main steps: carbon fixation, carbon reduction, and the regeneration of RUBP. First is carbon fixation. In order to make CO2 usable, it is fixed by an enzyme called Rubisco. Rubisco attaches CO2 to a 5-carbon molecule called RUBP, forming an unstable 6-carbon molecule. Because it is chemically unstable, it instantly splits in half, resulting in 2 molecules of 3-PGA. Next is carbon reduction. In carbon reduction, ATP from the light-dependent reaction transfers a phosphate group to both 3-PGA molecules. This phosphate group makes 3-PGA more reactive and ready to accept electrons. NADPH, also from the light-dependent reaction, donates electrons, reducing both 3-PGA, 2G, 3P. Before we move on to the last stage, you need to know that every Calvin cycle uses 3 CO2 molecules. So 3 CO2 molecules are fixed, making 3 unstable 6-carbon molecules. These split, resulting in 6 PGA molecules. Then, the 6 PGA molecules are reduced, creating 6 G3P molecules. In the last stage, the regeneration of RUBP, 5 of the 6 G3P molecules are converted into RUBP using ATP, allowing the cycle to continue. The last one G3P molecule exits the cycle to soon become sugar for the plant, whether that is glucose, sucrose, starch, cellulose, or other sugars. For example, one glucose molecule requires 2 G3P molecules, making the process occur twice for glucose to be made for the plant. Okay, so let's review. Carbon fixation occurs where Rubisco attaches 3 CO2 molecules to 3 molecules of RUBP to make 3 unstable 6-carbon molecule. The molecules then splits into half, creating 6 3-PGA molecules. Then, carbon reduction occurs, and the 6 3-PGA molecules gain phosphate from 6 ATP and electrons from 6 NADPH molecules, reducing the 6 3-PGA molecules to 6 G3P molecules. Last, the regeneration of RUBP occurs, and 5 of the 6 G3P molecules use energy from 3 ATP to regenerate 3 RUBP molecules for the cycle to occur again. The last one G3P molecule is used to make sugar.